Hi, welcome to my lab report where I'm going to tell you about a lab we completed a couple weeks ago where we actually transformed E. coli bacteria and made it glow. So transformation is a process where bacteria can take in DNA from their surrounding environment, and they do this by taking in plasmids, which are little bits of DNA. So this is what a plasmid looks like. The one that we used in this lab was called pGLO, and it was genetically modified, or that just means it was created by humans. So there's 4 parts to the plasmid that are important. Right here is called the origin of replication, which is where the DNA will start being copied. Over here is where it's resistant to ampicillin, which is a drug that kills E. coli bacteria. So in theory, if the bacteria were to pick up this plasmid, it would be able to resist ampicillin and not die. Over here we have the green fluorescent protein, which is actually taken from a jellyfish gene. So this is what should make the bacteria glow if it picks up the plasmid. There's also the arabinose operon, which is kind of like an on-off switch. So when the bacteria grows on the arabinose sugar, it triggers the green fluorescent protein to turn on, but it won't glow if there's no arabinose sugar. So what we did is we took 2 petri dishes where we grew normal E. coli bacteria. You can see here the dishes with the minus signs are where we did that. So on one dish, there's just lauria broth, which is this one. Lauria broth is kind of like a food for the bacteria. On this one, there's lauria broth and ampicillin, which, as I said earlier, ampicillin is a drug that kills bacteria. So on this side, we grew bacteria that was introduced to the plasmid. So on this one right here, we grew it on lauria broth and ampicillin. And remember that the plasmid is resistant to ampicillin, so once the bacteria picks up the plasmid, it should grow, which you can see there. On this one, the dish was covered in lauria broth, ampicillin, and the arabinose sugar, which is the trigger for the protein to glow. So for the protocol for transforming the bacteria, we started by adding a transformation solution, which was calcium chloride, to 2 different tubes. In one tube, we were growing, or it was just going to stay the plain E. coli bacteria. The other tube was going to be introduced to the plasmid. But we added the transformation solution to both. And that solution kind of helps the bacteria take in the plasmid more easily. So then we put the tubes on ice, we heat shocked them in a water bath, and then we put them back on ice before adding the plasmid to the tube. Oops. Yep. And then we spread the bacteria out. Oh, they're putting in the plasmid. And then spread it out on the petri dishes. So here you can see the growth we got. The first one has no growth because it's the ampicillin, and bacteria does not grow when there's ampicillin, so there's no growth there. On this one, we have growth because it's just the lauria broth. That's what the bacteria likes, so it grew. Over here were the bacteria that was introduced to the plasmid, and this one has some growth even though there's ampicillin, because remember the plasmid was resistant, so it grew little. There were some bacteria colonies. This is the plate with the arabinose sugar, ampicillin, and lauria broth, and you can see there's a lot of growth. And this is the plate which should glow green because of the arabinose sugar triggering it to glow green, the green fluorescent protein. So here we have all of the different groups and their plate with the arabinose sugar, which triggers the green fluorescent protein. You can see some of the groups are pretty successful in getting their bacteria to glow. This is my groups right here. So that was our lab. Thank you for watching.